Well, hello, everyone. My name is Pilar Alcaide, and I am from the University of Miami Miller School of Medicine. I have the privilege to be introducing one of today's speaker in the session that focuses around cardioimmunology, Dr. Han Zhu from Stanford University. Welcome, Han.
Thank you.
We're very excited to hear your presentation this afternoon at the cardioimmunology session. And could you please tell us, summarize for us, what are you going to be highlighting at today's session during your presentation?
Yeah, thank you. And it's an honor to be here and a pleasure to give this talk. So I'm the director of the Stanford Translational Cardio-Oncology program. So we're a bench-to-bedside program, and we pair closely with the clinical cardio-oncology efforts at Stanford. So our goal is to try to bring translational solutions to patients. And the way we approach this is by biobanking and then using single-cell multiomics, including single-cell RNA-seq, TCR sequencing, and protein immunophenotyping to identify culprit pathways. And then we bring it into mouse and iPSC models for validation. And ultimately, the goal is to bring a therapeutic solution back to patients in the form of clinical trials. And so in trying to do this for patients, we tackle the challenge of immunotherapy myocarditis, which is a serious side effect of cancer immunotherapy. Now, cancer immunotherapy is now being used in up to half of all cancer patients. Up to half of all cancer patients are now eligible to receive these drugs. And the way they work is by activating T cells against tumor. But unfortunately, there are some off-target effects. And one of the side effects is immune activation and autoimmunity against various organs, including the heart. And immunotherapy myocarditis is unfortunately still the most fatal side effect of immunotherapy, with up to a 40% case fatality rate. Very kind of serious barrier for patients to receive immunotherapy safely. And so what we've decided to tackle is to try and understand how T cells enter the heart to cause immunotherapy myocarditis. And in our prior work, we showed that cytotoxic CD8 T cells expressing this chemokine receptor, CXCR3, and you and I actually collaborated on this study. We showed that these T cells are recruited to the heart by CXCL9/10 secreting macrophages. And by interrupting this pathway, we can actually treat and mitigate the myocarditis. The next big question that my lab is now tackling is, how do we do this? How do we treat immunotherapy myocarditis without impacting tumor control? Because immunotherapy still needs to work, the T cells still need to enter tumor. And so how do we selectively target the heart while sparing the tumor? And so to do this, we've developed a tumor mouse model that also develops ICI myocarditis upon treatment with immunotherapy. And so in this talk, I present some exciting data that we have where we show that actually blocking CXCR3 both mitigates myocarditis and does not impact tumor, and may actually even have some beneficial effects in terms of controlling tumor. And so we look at this in both mouse and human samples. And so I'm really excited to continue to collaborate with you as well as other members of the Inflammation and Heart Failure Scientific Committee.
Well, I think this was an excellent summary of the beautiful work that you do.
Thank you.
I think the session will be very well attended, and I think your findings really set the stage for what can be done, starting from observations in patients, taking it to the bench side, and really figuring out the mechanism. And I'm personally very excited to see what you will be presenting in terms of the next steps of that landmark paper. And I think this brief summary of perhaps you have a way to really targeting traffic to one side and not without impacting the antitumor immunity is certainly like very, very exciting. So the next question I have for you is, what do you think will be the impact to those patients that now are receiving these combination therapies if your findings were being taken to the next step?
Yeah, I think the biggest thing is giving patients and providers the confidence that they can have a treatment for the side effect without impacting the cancer control. Because clinically, and I see cardio-oncology patients as well, and one of the questions that always comes up is, we've had this side effect, but if we give a drug, whether it's steroids or one of these other medications that we're using off-label, is it going to impact the long-term cancer control? Because that's really, one of the most important things for the patient. And then another really exciting next step that I'm excited about is actually predictive biomarkers. And so in the field, it's really been a little bit of a holy grail, like how can we predict who's going to develop the cardiotoxicity even before getting the therapy? And so one of the things, next steps that my lab is also tackling is if we take patients' blood pre-immunotherapy, are there ways that we can tell if someone's going to develop autoimmunity down the line? And so certainly there's a lot to tackle in this field and a lot of exciting opportunities. But first step is how do we treat the patients and give them confidence that the therapy is safe? And then the next step is how do we actually predict and even potentially prevent immunotherapy myocarditis?
That's exactly what was my question going to be next. Can we predict who will develop it? And I think that's why the session that we're going to have later today is really embraces all the interesting findings in the field of cardioimmunology that you are contributing to, and we will get more answers to what we can do to better treat the patients. So thank you so much.
Thank you for the opportunity to highlight my work.
Thank you, Han, and thank you all for watching.